previous speakers, I've tried to avoid using slides as much as I can. There's a few slides there. So that's why I have my notes, because otherwise I forget what I'm talking about. Thank you very much for the invitation to come here today to speak to you. It's always a pleasure to come to Italy, which is one of my favorite countries. And it's always a great pleasure to speak to an interested audience about your work. And there's few audiences as interested in our work as the patients and their families always are. As mentioned, my name is Alexander Smith, and I work in the Department of Genetics in the Institute of Ophthalmology and Moorfields Eye Hospital in London. And we are a group of about 50 researchers, medical doctors, biologists, who are trying to develop novel therapies for mostly inherited dystrophies. We do work on acquired disease as well, but most of our work is on inherited genetic retinal dystrophies. And our group was one of the first, or actually it was the first group to get to use gene therapy in the clinic to treat a form of retinal dystrophy, retinal degeneration. And that was, as mentioned before, was about 6 years ago, and that project has been going for that time, and we are about to finish that trial. And I will very briefly mention one of the results, just to convince you that gene therapy is actually possible in the clinic. But during those 6 years that clinical trial was running, we haven't been silent, we haven't been lazy. We have also worked on the development of other clinical trials, other retinal diseases, and one of those diseases is achromatopsia. And we are getting to a stage now that we hopefully will be able to start a clinical trial for achromatopsia. And in contrast to the previous speaker, we are working on CNGB3. We are getting to the stage where we hope to be able to start a clinical trial for CNGB3 in the next few years. I've been asked to tell you about our gene therapy work for achromatopsia, tell you basically what we've been doing and what we've achieved, and hopefully convince you that gene therapy for achromatopsia is not far away. But I know that we have got a mixed audience here, people with blue cone monochromacy, and unfortunately we as a group, although we work on a lot of diseases, blue cone monochromacy is not one of them. Nevertheless, at various points I will try to give you some ideas of what I think may or may not be possible for that disease as well. You have heard already an awful lot about genes and gene therapy, and there's a lot of overlap with what I was going to talk to you about. And I will nevertheless just have some overlap. I think a lot of people who are not biologists, who are not medics, don't really understand what genes are. And the way I'd like to look at it, what are actually genes is, and I think it's appropriate in Italy, I like to compare genes to recipes, like you use in the kitchen, a cooking recipe. A gene is nothing else than a description of how to perform a certain task. It tells the body, this is how you detect light, or this is how you make red blood cells, for instance. So it's a recipe, and like in the kitchen, if there's something wrong with a recipe, if it actually describes a certain ingredient wrong, or if the cooking time is too long, the recipe goes wrong, and what it ends up with, you can't eat. Well, if a gene is wrong, if there's a mutation in there, then the body won't be able to perform that specific task that the gene is supposed to do. And the severity of that What the result of that is, the disease that comes out of that depends very much on what the gene actually is. And we are working as a group on a lot of different retinal diseases. Some of them are very severe. We have recently developed a gene therapy for a gene called AIPL1. And the patients who have a mutation in that gene, they go blind within a few years. So then when they're 5, they have got no vision whatsoever. On the other hand, if you have a mutation in a gene called alpha transducin, you only have night blindness, and I'm sure night blindness is very unpleasant, but it's not the most debilitating disease ever. So having a genetic mutation can be quite terrible, can give you a very severe disease, but on the other hand, having a genetic mutation can also be very mild and unimportant. As mentioned, Achromatopsia has a number of diseases, a number of genes involved, and actually it's therefore not one disease, it's 6 different diseases or 5 different diseases. The main 2 forms are CNG-A3 and CNG-B3, and I will focus what I'm telling you about on those 2 genes. The reason why the 2 genes have such similar names is not a coincidence. Actually, they work together. They together have only one function, and that is that the CNG-A3 and B3 complex helps the cone photoreceptor cells turn light into an electrical signal. So the cells in the CNG-A3 or B3 patients can still detect light, that's fine. But once that light is detected, it can't turn it into an electrical signal that then goes to the brain for the brain to do its brain things. So it's very important that the cones are there and the cones can still detect light. And I'll come back to that later. Of course, in this audience, I don't need to tell you that what happens when the cones are not there. I'm sure you know better than I do what actually happens with poor visual acuity and photophobia. Blue cone monochromacy is in effect a slightly strange disease, and Dr. Cole has already explained that. It's not actually a defect in one gene, it is a defect in 2 genes at the same time. And in patients with blue cone monochromacy, both the red opsin, which detects red light, and the green opsin that detects green light is not either not there or it's not working. And therefore, the cones which are there, there's the cones that would be using the red opsin to detect red light. It's not doing nothing, it's not doing anything. There's the green cones which should be detecting green light. It's not doing anything. And those 2 cones, which are very important for your central vision, for your reading vision, are just sitting there, doing nothing, and slowly, as we mentioned before, some of patients have a slow degeneration where those cones die off because they just nothing for them to do. Boredom, probably. So, what now is gene therapy? What do we do now we know what the genes are and what those genes, which genes are involved in the disease? How do we actually treat it with gene therapy? Well, and the concept of gene therapy is very simple and very elegant. If we know that this particular disease is caused by this particular gene, and it should be in this particular organ or cell, then if we can actually put it back there, if we can put a normal, healthy copy of the gene back into the cells that require it, then hopefully we should have basically put the function that a gene describes back into the cell, and therefore the cell is healthy again, is working again. How do we do that? Well, with modern technology, and that's something from the last perhaps 20 to 30 years, it is actually fairly simple to isolate a bit of DNA, and DNA is the material that the genes sit on. It's like the paper that the recipe is written on. It's just a physical substance. We can isolate the right bit of DNA with the right gene. How do we get that into the cell then? Well, nature provides us with an answer to that. There is one type of organism in nature which is very good in putting DNA into cells, and just, you have heard it already, viruses do that very efficiently. So what we have learned to do over the past 15 years or 20 years is harness viruses. Take a virus, strip it out from its own dangerous, disease-causing DNA, put a bit of DNA in there that we are interested in, and then let it loose onto the, well, I would say patient, but actually mostly animal that we want to treat, and see can it restore the disease in the animal. And for us in the retinal field, there is actually only one virus which works really well, and we are lucky in that respect, because it is actually a virus called AAV, or adeno-associated virus, and AAV does not cause any disease. Just naturally, it doesn't cause any disease. We just heard we have got about 150 people here in the room. I'm going to guess that at this moment, about 30 to 50 of you have got an AAV infection. You just won't know about it because AAV does not cause disease. It shows this spreads through the body, dies out again, and the next day somebody else might get an infection. So that's an advantage, because the big danger of using viruses for gene therapy, of course, is if there's any virus in there which we haven't stripped out, is there any risk of causing a disease? Well, with AAV, there isn't. AAV is, fortunately, very safe. So now we have got a gene, let's say CNGB3. We've got a tool to get it into the cell, AAV. So how do we then go on developing a gene therapy protocol? For that, you need something to treat, and we are fortunate enough to have received from our collaborators in the United States a strain of mice which already lacks the CNGB3 gene. And so it's a mouse which, like an achromatopsia patient, has no functional CNGB3. It has therefore no cone function, and it has got poor vision. So these animals we could treat with a solution of AAV carrying CNGB3, and we inject that, as described in the lovely schematic earlier, we inject that underneath the retina and left those virus particles to infect the photoreceptor cells. And then basically the only thing we needed to do is follow the mice and see how they behaved, how do their retinas behave. So now I get to the slides, and I'm sorry, I apologize for the people who will not be able to see this. These are some sections, basically post-mortem sections of mouse eyes which have been treated with CNGB3 vector virus. In the bottom row, this is an untreated mouse, and over here is where CNGB3... Ooh, that is very dark, isn't it? So, it's on the screen, I can tell you. Basically, it is better, yes. It's not as bright as it should be, but it's good enough to see. Over here, this is an untreated mouse, and over here is where CNGB3 should be located. And as you can see, there's absolutely nothing there. We've stained this with a green dye, and if you now look at the other eye of the same mouse, basically when we inject these mice, you can see over here, there's all these green dots. That is where CNGB3 actually is now located in these mice, and it is where you would expect it, and it is where the amount we would expect to see. This is just to compare. This is all the S-cone opsin staining. So these are all the cones in this area. As you can see, the green signal and the red signal are approximately in the same area, so we can not only put CNGB3 where it is supposed to be, we can not only put CNGB3 back, but it's also going where it's supposed to be in the cones of the retina. So having put the CNGB3 back where it should be, does it actually work? Well, therefore we performed some electroretinography, ERG. As I mentioned earlier, CNG-A3 and B3 together work to turn light into an electrical signal. And so we can measure, we can flash light into the eye of these mice and see is there an electrical signal being generated. The top row here is measured under night light conditions. So these are the rod photoreceptor cells, and this is a normal mouse. This is an untreated mouse. You can see there's nothing wrong with the rods and the night vision in these mice, as you would expect. This is a photopic, so this is the daylight conditions, and this is a normal mouse. You can see lovely high ERG. High ERG is a good sign. In the untreated mouse, there is almost nothing there. There's a little bit, which in B3, in contrast to A3, in B3 that's normal. A little bit of signal there, but very little. And if we now look at the treated eye from the same animal, you can see the ERG. It's there, it's high, it's almost as high as in the normal mouse. So we can treat the defect in these mice very well. And this is a mouse which was treated at 1 month old. And if we actually do this ERG, which we can do time and time again because it doesn't actually do anything negative to the animal, it doesn't kill the animal, we can do this time and time again. And you can see that after almost 9 months, the ERG in the treated eye is still there. It's almost as high as in the wild-type mouse. So this is a single treatment at 1 month old, which 9 months for a mouse, that's a pretty long time. This is an almost lifelong treatment. And as you can see, it's still going. I mean, we could have measured after a year or maybe a year and a half and hopefully still find it. So this is one-off treatment that does not actually need topping up again and again. So we can actually restore the protein where it should be, or the gene where it should be. We can restore the function of the gene where it should be. Does this mean the mouse actually sees better? Well, we did not ask them to swim to colored flags. We had a different test. This is a test not for color vision, it's a test for visual acuity. What basically happens is the mouse sits in between 4 computer screens. And on those computer screens, there's black and white stripes which turn around as if the mouse is in a rotating drum. And the mouse has a tendency, or actually not just the mouse, everybody, human beings, mice, any large animal, has a tendency to actually try to follow the stripes with its head. So by making the stripes wider or narrower, we can test what the mouse can see because it's following, and as soon as it can't see it anymore, it stops following the rotating stripes. And so we can do that in mice, and we could do it again and again, so you get a very good measurement of what width of stripe the mouse can see, and therefore how good its visual acuity is. And again, this is a wild-type mouse with normal, for a mouse, for a human this is poor vision, but for a mouse this is normal vision. And actually in the untreated mouse, you can see there is a drop. It's not enormous. I mean, mice have very good rod vision because they are nocturnal animals and night animals, but there's a drop there, which we can restore the visual acuity with our treatment. The treated animals have a visual acuity which is almost normal compared to the wild-type mouse. So we can restore the gene, we can restore the gene function, and we believe we can restore visual function, at least in animals that are young. We have done the same test in animals that are about 6 months old, so that's middle-aged for a mouse. And in middle-aged mouse, we can't really restore vision. So we can restore the cones. The cones will work again, but we can't restore the vision. And that's probably not an eye problem, it's probably a brain problem. And that's something that we have generally ignore as eye people, but of course, in order to see, your brain also must interpret what comes from the eye into the brain, and a brain which has not had any cone input and no cone signals for a long time, for most of the life, the animal might not have been able to learn to interpret those signals. And that's something that, how that translates to the human situation, we'll have to wait and see. So we can restore vision. Can we also prolong the survival of the cones? As Dr. Cole has mentioned, there is a slow loss of cone cells in at least some, if not all, achromatopsia patients. It's also in our mice. And so we looked at the number of cones. I think it was the animal was 3 months old now. We looked at the number of cones in the treated eye and compared that to the untreated eye, and we can see that in the treated eye there are far more cones present. The red and the green signals here are the cones, and you can see in the treated eye there's more green signal here than there is in the untreated eye, and these are the numbers there. So we can also restore or at least stop the loss of cone cells there, and that's very important, and it's something that we need to stress. Gene therapy will only work if the cells that you want to treat are still there. The cones, they're not working in achromatopsia patients, but certainly for a long time the cones are still there, and as long as they're there, we can try to treat them. Once they're gone, and we can't do anything about that anymore. So basically, we believe, and I hope I've convinced you, that we can treat CNGB3 mice very efficiently. Sorry? Oh, yes. So yes, I'm convinced that we can treat CNGB3 mice very efficiently, and we hope that we can treat CNGB3 patients as efficiently. You might have noticed I did not address one major phenotype of achromatopsia, and that's photophobia. The problem with photophobia is that nobody really knows why patients have it. And therefore, I'm sure you want to know, can our gene therapy treat photophobia? Well, the honest answer is we don't really know. Because we don't know what causes it, and because mice don't have photophobia, well, actually, all mice have photophobia. Mice are nocturnal animals. They hate the daylight. So we can't test in our treated mice whether the treatment could prevent photophobia. So that, again, that's something we have to see. We have to wait and see, once we've treated patients, whether they report an improvement in photophobia after treatment. So that was all about CNGB3. You've already heard about the plans for CNGA3. What about blue cone monochromacy? Could this also work for blue cone monochromacy? Well, as I said, we don't work on it, but as a biologist, I don't see why not. I don't see why we couldn't also develop a gene therapy for blue cone monochromacy. Other groups have already shown that it is possible to put either the red or the green opsin back into cone cells and get it to work there. That would restore a form of bichromacy, a two-color vision. And more importantly, of course, it would reactivate cones that, assuming that sufficient number of cones are still there, it would reactivate cones that are not active in the center of the retina, and therefore I would expect that a gene therapy for BCM could also improve visual acuity in the patients. Whether we'll ever be able to put trichromacy, put both red and green back into cells and form trichromacy, I doubt that very much. So I don't think we'll ever be able to completely restore blue cone monochromacy to normal vision, but I don't think that is probably not the main aim that people here would want to achieve. Thus far, I've only talked about what we've done to mice, and that's basically because for achromatopsia, we've only worked in mice. Still, as I said before, we have been doing a clinical trial for a different form of retinal disease. It's a disease called Leber congenital amaurosis, or LCA, and it's caused by a gene called RPE65. And patients with RPE65 disease, they are night blind from birth, and they lose their daylight vision in their 20s. So after 25-ish, they are legally and practically blind. And this was disease for various reasons I won't go into. It is a disease we wanted to treat first because it was a very good candidate for testing. We looked at safety primarily, of course. Nobody had ever done this before, so we had to show that it's safe to do. And fortunately, we, and there's 2 other trials in America have been occurring since, have not found any major safety concerns, nothing that would stop us going onto a next trial for a different disease, which is good. We also looked at a huge number of different retinal assessments, measurements of retinal function, and I won't go through them all. I will just show you the one striking result that we found. And that is not working after all. But I have another plan, and that is... Okay, so this is a patient who we are asking the patient... That's not working at all anymore, is it? Yeah. In our view, that was the first proof that gene therapy for retinal disease is not only possible, but it is actually working. There is a lot to be gained from it. And this trial has been a lot of hard work. But we've learned so much, not just about this disease, but learned about doing clinical trials, how to get through the hoops, how to treat patients. And all that information now will help us with the CNGB3 clinical trials as well. Recently, we obtained funding from the Medical Research Council, which is the organization which distributes the government money in England, to do a clinical trial. It's about £3 million worth of funding to do a clinical trial for CNGB3. And at the moment, we are just finalizing the final details of the virus and the gene, how we want to compile it all in one vector. And at the end of this year, we intend to make a virus, a vector, to the standards that are required, the very high standards that are required to be able to use them in people. And once that is done, hopefully, that's the planning, by the end of 2015, so autumn 2015, we are hoping to start a clinical trial for CNGB3. And with that, I would like to thank the organizers again and thank you all for listening. And if you have any question, please ask.